Welcome to the BIRCH Bioinformatics system. My name is Brian Frostensky, and I'd like to tell you about the bioinformatics tools that my lab has developed for many of the important tasks people want to do, in particular genomics tasks and standard sequence analysis tasks, working with BLAST databases. The list is quite large. One of the important things is that BIRCH ties together all of these tasks in a family of graphic interfaces using the BioLegato application that we'll talk about a little bit more in a moment. So you can best think of BioLegato as a program that runs other programs. Each BioLegato application is able to display a certain type of data, whether it be sequence alignments or BLAST results or lists of sequencing read files, and then do various tasks on them. So each BioLegato application is specialized for a particular type of data. But one of the powers of BioLegato is when BioLegato runs one analysis step, usually the output can pop up in a new BioLegato window specific for that type of data. And so this allows you to chain together many different tasks one after another and experiment as you go along. We call this process ad hoc pipelining. And to illustrate that, I'm just going to go through a few quick examples to show you how it works. So as an example of how ad hoc pipelining is both powerful and flexible, let's just take the example of finding out something about a class of proteins that I'm interested in, originally cloned in P as DRR206, defense proteins that are activated by pathogens, and later also cloned independently as dirigent proteins, guide proteins that help in lignin formation. And so here we have a query builder in the BioLegato application BLNCBI, and you'll note that we can set query terms, like for example, if I want to find proteins of this type in the flowering plants, I can set the organism field to Magnolia phyta, and then add an AND clause so that we can have two search terms in parentheses, either the text word matches DRR206 or the text word matches dirigent, and I have parentheses that I can put around the expressions to make sure they're evaluated properly. And so once I've set my search parameters, BLNCBI will retrieve those, or at least references to those sequences from NCBI. And so it looks like all of these are sequences from P, so we didn't get a lot of them initially as hits, but if I want to find more examples from other species, I can actually retrieve the sequences themselves now using protfetch, and that will return the output to another BioLegato application called BLProtein. So BLProtein is a BioLegato application for protein tasks. You can see that you select sequences and then you can do various tasks on them, pairwise comparisons, database searches, pattern searches, etc. We'll just take one and show an example of doing a BLAST search. And in this case, we'll do a tBLASTn search on the non-redundant nucleotide database. But instead of searching at NCBI, we happen to have a copy of this database on our local system, so we can run this search and search the local database and then get the results. And I'm going to edit out the time it takes to actually run the program, and then we'll come back when that's completed. So now we have our results back in two windows. One, you can see the usual web page with the BLAST output that lets us take a look at just about any of the hits, and then very conveniently a table of the same hits with all of the same information but in table form. And you can see there are 278 hits. The table is really useful because you can do spreadsheet-like things with it. For example, if we have a look at the sequence length field, we'll see that some of these are really very long sequences, probably genome fragments and if we want to just work with gene size sequences, what we can do is sort them, and here is an example where I've sorted them based on the length of the sequence. So now I've selected only the gene-sized ones, and I've left behind the back clones and chromosome fragments. So if I want to get these sequences from NCBI, I run seqfetch, which will retrieve the DNA sequences into a BLDNA window, which is yet another BioLegato specifically for DNA sequences. So now you've got the basic idea behind ad hoc pipelining. For every step that we do, the output comes to a new BioLegato window specific for the type of data that we're working with. So we can quickly go from one step to another step and never lose the ability to keep working with data. So here, as you can see, of course, we have a lot of different sequence tasks that can be run on the raw DNA sequences we've retrieved. If I wanted to get a multiple alignment to the proteins, then what I really want to do is run BioLegato to get the coding sequences extracted, and then now having the coding sequences extracted, I can run ribosome to get the actual amino acid sequences, and then use MAFT to get a protein alignment. So the protein alignment, there we see it in the BioLegato window, and I can either work with that, for example, make phylogenetic trees out of the alignment, or even send it to a program like JALView, where I can examine the alignment in more depth, looking for conserved regions, or how the sequences group using principal component analysis. And so the ad hoc pipelining lets us do many different things at each step before deciding then what do we want to do at the next step. So where BioLegato can really help us is with genome and transcriptome tasks. And these are difficult, first of all, because the programs are difficult to install. Each program has a tremendously difficult learning curve, and usually creating the input files so that you have the files paired up with forward and reverse reads, that by itself is a big problem. And even one trivial thing, seemingly trivial, is just that the names of the files are long. So at the bottom here you can see that we have Illumina forward and reverse read files with these very incomprehensible names, and one of the tools in BL Reads, which is the BioLegato application for working with sequencing reads, is simply to create symbolic links to the original files with shorter names. So if we simply replace the or shorten the name of the symbolic link by getting rid of the beginning part of these file names, we can create a very orderly and understandable set of symbolic links that can be used as aliases for the files with the actual long names. So here we see there are 2 time points, 18 hours and 24 hours, 3 replicates, and then the R1 and R2 files are the left and right reads. So having done that, then we can run programs. Just to show another example, here we've run the FastQC program to generate HTML reports, and so that was just done by selecting all of these reads and then running FastQC from the FastQC menu. And here is the output. If we wanted to see an example of FastQC output, we get all sorts of information on the quality of the read. So here, for example, is a graph showing the quality of each nucleotide, or the average quality of nucleotides at each position among hundreds of thousands or millions of reads, depending on how big a data set you're working with. And this allows us to evaluate the quality of the reads that we're working with. Well, once you have that, you can go then to these symbolic links and run programs like Trimomatic to get rid of the adapters. The sequencing adapters have to be removed. And in order to do that, we need to be able to tell Trimomatic which files belong together as forward and reverse reads. So for this purpose, we've got the guess pairs tool. I'm telling guess pairs here that R1 stands for forward direction, R2 stands for reverse direction, and then match files that have the file extension fastq.gz. So here's the output from guess pairs. It usually guesses right, and we could then run any program on these pairs of read files. So if I want to run Trim-O-Matic, of course, there is the menu for running Trim-O-Matic, and we already have the Trim-O-Matic results ready to look at in the next file in the list here. So Trim-O-Matic will separate out the reads based on whether both reads in a pair, forward and reverse, are present. Those files end in a P, and if they end in a U, that means that there were unpaired reads. You're missing one of the two reads, and so those missing reads can often mess up assembly programs. So generally speaking, we want to avoid working with them, and instead what we'll do is we'll use guess pairs to just get the files that end in p.fastq so that we can work with those. So I guess I have to select all before I run that, and we'll rerun guess pairs, and there are the reads that we want to work with. And so these reads now are ready for read correction. And so now that I've got the paired reads from Trim-O-Matic for the trimmed reads, I can correct the reads using rcorrector. So this is a program that is specific for correcting RNA reads. And so I've already run rcorrector, by the way, so I happen to have the output for rcorrector, and that is there we go. There is the output for rcorrector, and now all of the files have a .cor.fq file extension. So we're ready now to do an actual transcriptome assembly, and the transcriptome assembly is done. We have a choice actually of three assembly programs: RNA Spades, SOAP de novo trans, and Trinity. I've got the output here from Trinity, and so you can see, for example, if we open up the Trinity log file, there is a running transcript of the process of assembling the transcriptome. And then the final step, of course, is to analyze the quality of the result using transrate. So I'm going to open up the transrate directory where the output was saved, and then that has two important files in it. One is the transrate log file, which, if we look at that, it gives us a report with statistics on the metrics of contig assembly, how big the contigs are, and how many had an open reading frame, that sort of thing, plus also how well did the reads map to the transcripts. And then finally, a final assembly score that tries to put together all that information into a single number. Now we also get that same result, that same set of results, in a spreadsheet file. And the reason that the spreadsheet file is really useful is that it lets us compare assemblies. So I have another spreadsheet file over here that shows these results pasted in after doing RNA Spades, Trinity, and SOAP de novo trans, so we can compare the quality scores and how well the reads map and so forth. We can try rerunning the programs many times with different parameters and see which is the best assembly. So the last thing I want to cover just very briefly is to show how BioLegato can help you install and manage local copies of BLAST databases. And sometimes it's very useful to be able to have your own copies of the BLAST databases and do your BLAST searches in-house. So the BIRCH Admin administration tool is yet another BioLegato that lets you do various tasks, including looking at or managing your BLAST databases. So I have several installed here, and I'm just going to click on a button that will open up a spreadsheet that shows which databases I currently have installed. And so you can see that I've got the non-redundant nucleotide, SwissProt, and the PDB three-dimensional protein amino acid database along with TaxDB. If I was interested in installing some additional databases, first of all, I have to see how much disk space is left. In this case, I've got 350 gigabytes of disk space left. And so if I want to see the other possibilities that I could install from NCBI, I now will get a report on the databases available for FTP download. And so there's a list. For example, the RefSeq gene would be 181 gigabytes. RefSeq RNA would be, well, I guess that's the uncompressed, or the compressed. The final RefSeq RNA would be about 22.9 gigabytes. So I have an idea of which databases I might want to download, and then that would allow me to go back to the admin tool and actually add databases. So as you can see here, for any database that is already installed, like the non-redundant nucleotide database, we have a plus in the window for, or in the menu item for the database. If it's not installed, there's a minus. If, for example, I wanted to install the 16S microbial database, that's a pretty short one, then what I could do is click on Add and then Run in order to install that database. I'm not going to do that now. I just mainly want to illustrate how easy it makes the process of managing your databases, because a lot of it really is about, first of all, the difficulty of downloading and uncompressing the data, and then also managing your disk space. And so that's one of the things that BioLegato does for us in this process. You can learn more about BIRCH at other videos on our YouTube channel, and also by going to the BIRCH website shown here to look at the tutorials, which go into much more depth on each of the different types of things that BioLegato can do, as well as letting you download a copy of BIRCH on your own system. So thank you for watching, and I hope you try the BIRCH system.